biotechnology. Today we're going to think about how to separate DNA and how to separate other parts of the cell material. So assuming we know what biotechnology is and that we're using a definition where we're getting some organism to make a protein or a chemical that we want, we now need to get that chemical or protein out of the cell. It might be DNA, it might be a useful protein, it might be an antibiotic, but our problem now is that we've generated it inside some cells, and that most of the things inside the cell are not what we want. As you probably know from normal life, over 90% of all of the material in the cell is water, but then even if we dried it, still over 90% of the material is other things, not what we want. So taking a simple example of extracting DNA from a cell, so we're only interested in getting the DNA out of the cell and keeping it for some other use. We will do this in the lab later, but today we'll look at it theoretically. So we want to take this DNA here out of the nucleus of a cell. Let's imagine it's one of my cells. So we've taken a cheek cell sample like they would do if I was maybe a suspect in a murder, and we want to get this DNA and extract it so that we can do our test later. So what are we going to do? First of all, we need to get rid of all of this outside cell material by breaking it up. So we need to look at how the cell wall is made up. Because we're talking about animal cells here, human cells, the cell membrane is a phospholipid bilayer, which you all know from school, but you might not know what a phospholipid really is. So here it is. This is glycerol, so propane triol, and all of the hydroxide groups on it, the 3 alcohol groups on it, are esterified. So this is the ester of a fatty acid, this is the ester of a fatty acid, and this one is not. This one is the phosphate group. The phosphate group here, phosphor, with 4 oxygens attached to it, and one of them is negatively charged. It's not always the same one. This negative charge can push in, knocking one out to one of the other oxygens. So the negative charge is delocalized across all 3 of those oxygens. That makes it relatively stable, makes it also comparatively acidic, so it doesn't get protonated very easily. This has a structure very much like a soap. So if we have a soap, we've got a water disliking region, a hydrophobic region, which in reality just doesn't attach to water as well as water attaches to itself. So let's be clear and say it properly. This region, the ester bond is not very polar, so it doesn't dissolve in water very well. But this group is charged, and so that is the bit that goes into the water. When we draw it as a ball with 2 legs, the 2 legs are these groups, and the ball at the top is this group. It's comparatively big because the charge on here repels the next one, so it behaves as if this group is actually quite big. It's actually surrounded with water, and the next one can't get that close. The easiest way to get through this would be to use a blender and just cut it up, but we can help that. So we would like to help the process along a little bit, and one of the ways that we could do this is to add some soap. So you may notice that I've added a hydrogen here because I got the wrong number of bonds to the phosphorus. If we look up to this one over here, the sodium dodecyl sulfate, SDS, or the sodium lauryl sulfate, or sodium laureth sulfate, as you will often see on personal products. You will see there are 12 carbons in this chain. Then there is a very similar structure, but as sulfur has a different number of bonds to phosphorus, the bonding here is slightly different, and the sulfur group has a negative charge. The sulfate group has a negative charge, leaving it very soluble in water. This is the most common soap in our modern world, with either a sodium, most often a sodium, sometimes a potassium ion attached to it, because it needs to be 0 charge if we want to move it around, dissolve it in something else. What we often see it in is cleaning products. So shower gel is a good example, liquid soap, but also in toothpaste, for example, there's sodium dodecyl sulfate. It's the foaming agent that makes it, that fools us into believing that it works. What we're going to do is we're going to mix this with our cells, and the soap will get in between these groups, behaving like one of them, but instead of forming a double layer, it will tend to make a hole in it and let the insides become outsides, which is obviously not great for the cell. This other soap that I've drawn just over here is actually better, but it's quite expensive. So why is this one better? It's better because it's a positively charged soap, so that group there is effectively an amine where the hydrogens have been replaced with methyl groups. That means it can't lose them, which locks on this positive charge on the nitrogen. So the nitrogen is allowed to have this group. It would normally be NH4+, for example, but because we've got methyl groups instead, or we've got and the chain, it can't get rid of its charge, and so it's stuck in that form. The counterion in this case is bromide, and this is commonly known by the name CTAB, cetyl trimethyl ammonium bromide. It's particularly interesting at the moment because it's an antibacterial, antiviral agent, because we discover that most of these compounds, most cell wall components, including sugars, so including bacteria, including viruses, most of them are negatively charged, and that means a positively charged soap is actually better at messing with them. It's also better at messing with us, so it damages our skin. So now what we've generated is effectively soup. We've got the inside of the cell and let it all out, and we've now got a mixture of proteins and sugars, different lengths of sugars, phospholipids, that was the cell wall, but also fat deposits, DNA, RNA, and anything else that was in the cell, so salts and things. However, now that we've done this, we've committed a crime and we have to do something very quickly. So what is the problem? This is an animal cell, it was me, and normally in an animal cell there is no DNA outside the nucleus. So what the body knows is, it doesn't know it really, it doesn't think, but there is normally no DNA outside the nucleus of an animal cell. However, if a virus attacks us, it will inject DNA or RNA into the cell. If it's RNA, it's a little bit more difficult to deal with, but if it's DNA and it's hanging around in here, it's not supposed to be there, so there's a very easy way that the cell can defend itself against that, and that is by having an enzyme in there to cut it up into pieces. So it will have an enzyme called DNAase, the A's meaning to break, DNAase, so an enzyme that breaks DNA, and it just sits in here and waits and does nothing until a virus attacks, and then it cuts it up. However, it's going to be a problem for us, because the moment the DNA gets out of the nucleus, and the nuclear membrane is no different from the outside membrane, so that is going to break at the same time. The moment that they mix together, the DNAase is going to start killing our DNA, cutting it into little bits, which is obviously not what we want. So what we're going to do is add a protein that will break down proteins, sort of traitor to its kind. That will be a digestive protein that will break down proteins into smaller sections so they don't work, and it will break specifically the DNAase, whose normal job is to stop DNA from getting into the cell. The protease has another function actually. Normally when DNA is present in our cell, it is not as an unraveled chain of spaghetti, because if you imagine a sewing kit full of wool or spaghetti in a bowl, it's very difficult to find the piece of spaghetti that you need, or to unravel the wool to get to the bit that you need. So the body has a more effective system. What it does is it wraps the DNA around effectively spools that are made of protein, so special proteins that hold the DNA in a neat form so that the bits that are needed are more accessible. That is also degraded by the protease. In our case, we have to heat it up. Just make some disclose where I'm getting it from. So I'm getting a kit from a company called Biorad. It's a relatively big life sciences company, and they make educational kits which are very handy. And so I'm going to use one of those, and it uses a type of protease that you have to heat up a little bit, which is practically useful because it means it doesn't degrade itself at normal temperatures, so it's a lot easier to handle. Because the problem with proteases is obviously that they are a protein that breaks down proteins. So if you keep it in a bucket for a while, you will end up with nothing, or one molecule left eventually. Then we get on to the thing that I've written as a problem. So we have our DNA, and DNA, as you know, is a chain of sugars, but with a phosphate backbone. So we call it a phosphate backbone. So each sugar is these rungs in between. That's two sugars next to each other. And down here is a chain of sugar, phosphate, sugar, phosphate. The problem is every phosphate has a negative charge, mostly. Obviously, because there are a lot of them and they're connected to each other, some of them will be protonated and not be very charged, but on average there is a huge amount of charge on this molecule, and it's all negative. That means that it's very difficult to get this out of water, because there's a big repulsive force between this large negative charge and this large negative charge, and they all add up to make it very difficult. The other problem is all of the other parts of the soup, the protein, the amino acids that we've generated, or small chains of protein that we've generated, so polypeptides that we've made by cutting up all of the inside of the cell, all the bits of sugars, all of the RNA, all of the salts, everything is still in our soup. So all that we've done is saved our DNA up to this point. Because we've cut the protein up into small bits, because we've broken the cell walls up completely, we now have an opportunity. The DNA is the largest molecule left, but it isn't really soluble in water, so we need to think of a way to get it out. Now we need to turn to chemistry again. So from chemistry, what we know is this is going to be really soluble in water, but if we change it to another solvent that is not as polar, it won't be as soluble. However, we're going to need some positive ions to go in between this gap, otherwise it will never crystallize. So what can we do? We can increase the salt concentration. If we increase the salt concentration, let's say sodium chloride, so let's have some Na plus ions in between, then we can stack these plus minus, and that is then allowed. It will retain quite a lot of water, but that will still allow us to separate this with water in it from all the proteins and other stuff in the cell. Then what we're going to do is we're going to convert this into a less polar solvent, so this water into a less polar solvent, and we're going to make it unhappy by cooling it down. So this is the Gibbs free energy equation from physical chemistry, but it tells us what we need to know about this process. So if we have a process where generating a solid is going to make it more ordered, then we will need to reduce the temperature to make that process more likely. Assuming we will also have to set it up so that we get some energy out from this, and that is caused by the different components interacting with each other, so these positive and negative ions interacting with each other, and the water and whatever we're going to add to make the water less polar. So what can we add to make water less polar? It has to be something that will mix with the water perfectly, because obviously if we throw some oil in, it'll just hang around on the top. And it has to be less polar than water. A good example for that would be ethanol. So ethanol is a solvent that's less polar than water. Salt won't dissolve in it very well, and it will mix with the water and make it in general less polar, so the total water polarity, the total solvent polarity will go down. It's slightly better in my experience if we use isopropanol, because isopropanol has more carbons on it and it's cheap, and it doesn't interact with any of the things that we don't want to destroy. The problem usually is if we want to retain a protein and we want to retain its structure, alcohols will mess with that because they will interact with the hydrophobic parts of the protein and cause it to unravel. So now we've extracted some DNA, maybe we should think about what we might want to do with it. So going back to the example of being suspected of doing a crime, we will want to know, is this person the same as the person who left a sample at the scene of the crime? So what do we know about DNA? Well, if you've had a look at the other videos in here that I didn't make but I linked to, you will know that we have lots of DNA arranged in chromosomes, and that most of the DNA is identical between people. The only real difference between people is the sex chromosome, which is the biggest and most obvious difference chemically. If we look at different races or skin color or eye color, those things are very, very difficult to find in our DNA, and very often the genes that code for them are present in both types of people that have more or less of whatever it is, hairy arms, brown eyes, blue eyes, whatever it is. The genes are present, just some of them have a slight difference, and so they don't function exactly the same, or they're just switched on or switched off. So the coding parts of the DNA are not what we want to look at. But coming back to our current virus crisis, when we're infected with a virus, it inserts sometimes its DNA into our DNA. So it cuts somewhere inside our DNA and adds some data. So let us have a look at that in the case of the HPV, the human papillomavirus, which causes cervical cancer, obviously only in people who have a cervix. So HPV is a virus that attacks us, and it attacks skin cells particularly, or types of skin cells, and it inserts its genetic material somewhere in the cell. The problem is that it inserts it randomly somewhere along the genome, and most of the time that will be fine because it will do nothing at all, or it will be in a gene that's not currently working. So maybe it's a skin cell and it's a gene that codes for making bones, so it will not affect the cell at all. Or it's just between genes, or it's doing something else. But occasionally it will do something important, so it will interfere with an important function of the cell, and then the cell will mostly die, which for us, our human body, is not super bad. Obviously the virus doesn't get to survive that. It might float away, but it will probably be cleared up with the rest of the cell. So that is not a great sort of solution for the virus, but it is cheaper from an information point of view for it to just shove the data in randomly. Most of the time it will work. The problem comes if we have a persistent infection. So this virus is hanging around inside the body, occasionally infecting cells, because eventually it will do something really bad. So what it's eventually going to do is it's going to affect one of the mechanisms that the cell uses to detect that it is doing something wrong, that it is infected, and that is obviously bad. It's a pre-cancerous stage. Why is this important? It's important because there are other types of viruses which also do the same thing, but they're a bit cleverer. They insert their DNA between genes or inside the non-coding DNA inside genes. So if we take a gene in an animal, it has domains inside it, so pieces of genetic material that don't do anything, or for the purposes of that gene they don't do anything, and they get removed before it's used to make protein. The virus then can insert its information inside these sections and nothing will happen. At least it won't get noticed and it won't kill the cell. That, for the purposes of the virus, is a good result. For the purposes of the animal, the human. It's not that bad. It's now infected with a virus, which is a problem to deal with, but it's not going to give it cancer. Obviously, for genetics point of view, the parts of the gene that code, or the parts of the genome that code for proteins are the most important. However, for the purposes of telling the difference between individuals, it's the non-coding parts that are more important, because those are the ones that will vary the quickest between generations. Those are the ones that are different between brother and other brother. Obviously, telling the difference between a brother and sister is comparatively easy. So we could generate an assay, a test, that will look for specific sequences inside the DNA, but that's not super effective because all of these regions contain repeats. They contain the same information over and over again, just a different number of them, because that's what's inserted as a virus comes along, puts its information in, and the body tries to repair it, so cuts it back out again, and little bits are left over, and those are longer or shorter in all of us. So what we're actually interested in is the length of that, and so we want a technique that will measure the length of things. So therefore we need gel chromatography. So what do? We take a gel block. It's full of water. Let's think about what else will be in there in a moment. We're going to apply a voltage to it because our DNA, remember, was negatively charged. It's got a chain and it's got negatively charged parts on it. Now we'll discover something. All the DNA has the same negative charge per unit length, so there's a problem there. We can't tell the difference between differently charged groups because they're all charged the same. So our DNA is now going to move under the field through a gel. Because the gel is effectively a very viscous medium and it's full of particles that the DNA has to get past, the longer parts will go slower than the short parts. Therefore, we need a method for cutting the DNA into sections so that we know what they are. Because if we take a whole chromosome, it's so big that the differences between people is very minor and we won't see it in this measurement. Luckily, we've mentioned this already, there are chemicals, there are enzymes inside the cell body that are specially there to attack DNA and cut it into pieces. A great many of these cut the DNA only at certain sequences, and that's really useful because that means that if we mix them together we can cut DNA at the same place for the same person. So if we take a bunch of DNA, it will all be cut to the same lengths, and we will have a mix of lengths and be able to separate them. So when we turn our electric field on, there is a force on the DNA molecules due to the electrical field that drags them in this direction, that forces them in this direction, and there's a drag on them, which is the viscosity of the liquid and the gel. If we did this in a liquid, it would move as a blob up and push the liquid around with it, so we need the gel there to prevent the chemicals from being able to diffuse together. That would cause a type of convection, type of electrostatic convection, so we would pull our particles all up together and then we would get no separation at all. So what we need is the gel in there to prevent that. It also helps to stop the DNA from diffusing out and getting somewhere else. You'll notice I've drawn two bars there. That's because there's a problem with this technique. There's a problem with all techniques. This one is just the same. The problem here is we apply a voltage here. We've got a reasonably high current, so we get a warming effect. We will heat up this material, and that will change the viscosity of the water, and that will mean that the DNA will move faster and faster as time goes on. So it's therefore very difficult to standardize the temperature and the conditions that we have. To avoid that, we always run an internal standard, that's some DNA that we know the length of. We will get a set of bars, a set of measurements. So this is our DNA, these are different components of the DNA. We will get a set of measurements that we can use to determine our actual sample. And if it runs warmer, then these will move further, and we have to turn it off earlier. If it's colder, then they won't get as far. But if we stretch and compress all of the features using the standard, then we can compare two tests with each other and it works properly. A couple of asides because I missed some things. First of all, if we apply a voltage to water and it's high enough, we run some current through it, what we would expect to see is that the negative electrode will make hydrogen from H+, so we're taking H+ away. We'll also make hydroxide ions from water. Effectively we're making new H+ when it runs out. And on the other side, the positive electrode, we will make oxygen from OH-, and eventually we'll have to make more. So we'll have to make more OH-, which leaves H+ left over. That will have the effect of making this electrode very acidic and this electrode very basic. And therefore the liquid in there, which I said I was going to mention, the liquid in there has to be a buffer to prevent the pH from changing, because what we don't want to happen is the DNA to change charge while we're doing the process. That would suck. On the other side, you may have remembered from polymer chemistry a different type of chromatography called gel permeation chromatography, and that works in exactly the opposite way, extremely confusingly. That confuses me, possibly confuses you. I had to look it up again today to remind myself which one we're looking at here. This type of chromatography is different because it contains effectively beads, so we have a column with beads in it, and inside the beads are small pores that the molecules can get lost in. What this means is if we have a big molecule coming along, it can't get in, and so it doesn't go inside, and so it passes straight through the column and goes out the end. If we have a small molecule, it can go inside and get lost. The medium-sized molecules struggle to get inside, so they don't go very much inside. We're measuring effectively the amount of time that the molecule spends inside the bead, where it's not moving, and the amount of time that it spends outside the bead, where it's moving at the speed of the liquid. The end effect of that is exactly the opposite of what we have here, that the long molecules, the larger molecules, go through faster, and the shorter molecules go through slower. That is what I find confusing about those two techniques. So if we take the whole process, we have to extract DNA from the sample that contains a lot of other things, cut it up with restriction enzymes. So I've given you the name now, restriction enzymes. That is the type of enzyme that cuts up DNA, usually bacterial ones, because bacteria also have to protect themselves against viral attack. Then we carry out a gel electrophoresis, which is the name for that technique. Electrophoresis means we are moving things under electrical charge. Gel is the gel, and then we need to record it somehow, which we haven't talked about. So obviously the DNA is a chemical that is fairly transparent, and if we get very little of it, we're hardly going to be able to see it inside a polymer gel with some buffer in and holes and all kinds of things. So we need to label that somehow so that we can measure it or record it in some other way. So one way would be to attach radioactive groups to our DNA, and then we'll be able to measure it with a photographic film. But that has fallen out of favor because obviously it generates radioactive waste, which we have to deal with, and low-level radioactive waste is super expensive to get rid of and very annoying. So instead of that, we would usually attach a dye to it, either a normal dye so we can see it in a photograph, or a fluorescent dye so that we can see it by shining ultraviolet light on it and we get another color back out. Another possibility would be to transfer the DNA from the gel onto another surface and then to color it there. That would be called a blot. Because we've mentioned gel electrophoresis, we probably should also mention how it can be used for proteins as well, because we might more usually be trying to extract a protein from cells. So if we want to use this technique on proteins, we have a new problem. As you know from biochemistry, proteins are a chain of amino acids, and on the side of the amino acids there are various groups, some of which are acid groups like this, that can deprotonate to form negative charges when in the basic conditions, and amino groups such as this that can protonate to form positive groups in acidic conditions. That gives us a mixed positive and negative charge all over the molecule that varies depending on pH. So if we look at the charge to pH, we can see that at some pHs, at low pH, it's typically positively charged, at high pH it's typically negatively charged, and at some pH it will have a net zero charge, although there will still be positive and negative charges, they just balance. The point of zero charge, the pH of zero charge for each molecule, so that is often called PZC, or the isoelectric point, iso being the same electric charge, so it's got the same positive and the same negative charge. That will vary for different proteins, so let me draw a couple on. So now we've got 3 different proteins and they've got a different point of zero charge, a different pH where they would have no charge at all. So that obviously gives us a problem. We can't do the same type of gel electrophoresis as we did with the DNA because we don't know what the charge on our protein is, we don't know what the charge per unit length is, we don't really know very much about our protein. So we have to do something. One of 2 things. The first possibility is that we put some soap in, SDS, sodium dodecyl sulfate, just like we had before, and that does some magic, something really cool. If there's a positive charge on the protein, so the parts of the protein that have positive charge on, the SDS will stick to, and reverse the charge to make it slightly negative. If there's no charge on the protein, and it's slightly hydrophobic, the SDS will stick to it and give it a slightly negative charge. And if there's negative charge on part of the protein, like down here for example on this one, then it won't stick at all, giving an average charge that's pretty much constant over the entire molecule. There is a problem, however. The soap will tend to denature the protein molecule and cause it to open out, which I've drawn like that. That's good because we want to measure the length of the protein molecule. It's bad because we won't be getting it back in any functional form, not very easily anyway. So now we can do our electrophoresis exactly the same as we did before with the DNA, and we will get a measurement of the length of the protein, how many amino acids there are in it, directly from the same measurement. We will also have to do a comparison measurement with a standard because of the same problems that before, that the gel is difficult to get the same every time and that the temperature varies during the measurement very often, so we need a standard. The other possibility is even more awesome. What we can do here is we leave the protein as it is, and we're interested in the pH at which it has zero charge. So instead of having a strong buffer inside our gel, we have a much weaker buffer solution, and we set up a pH gradient that is made up almost automatically from the current that flows. It's much better if you make it beforehand, but it will automatically generate a pH gradient over time. So we have at the negative end a high pH and at the positive end a low pH. Because the molecule has no charge at its point of zero charge, there is no force on the molecule at that point. So if we take this red one, which is relatively basic, if it moves slightly down, it gets into a more basic region, it gets slightly more negatively charged, and then it gets pushed back upwards, in this case, to the red dot. Maybe it goes past, so it goes past, it becomes protonated, gets more positively charged, and then it gets pushed back to the exact place it was before, and the protein can stick there. Up in the middle the same happens, so our other protein that has a different point of zero charge will become stuck here, and the protein that has an acidic point of zero charge is stuck up here, the green one. That's brilliant. It has lots of advantages. Major advantage is that we can wait for longer because the protein can't escape. If we do this electrophoresis, if we forget, then it goes too far and the measurement is ruined. There's nothing you can do to get it back, or not very easily. If we leave this one on, nothing happens. The protein is stuck where it is. Of course, it can move sideways, so it will drift sideways, but it can't escape that point where it is the height where it is at zero charge. This technique is called isoelectric focusing, and this technique is called SDS page. I've ringed the two things that we know about. So the SDS is SDS, that's the surfactant, and GE stands for gel electrophoresis. Okay, that's enough for one time. I'll see you next time, or in reality I won't see you because I can't see through the lens, but you'll see me next time.